Welcome back. Today's lecture is going to bring us to the first of 7 lectures on the nervous system. This is an extraordinarily complex, highly evolved system, certainly the most evolved of all species, and one that's going to be very difficult to cover in just 7 lectures. But we're going to look at, again, the structure and function, the coverings, the fluids, the exchanges, and the connections of the nervous system from top to bottom, and hopefully give you an understanding of how the brain works, how the special senses protect us and regulate all the other systems in the body. The brain is about one of the largest organs in the body. It weighs more than 3 pounds, contains more than 100 billion cells, if you can imagine a number like that. It only constitutes about 2% of the body's weight, yet it receives 20% of the blood supply. So we know the importance just by that figure alone. And basically, all the other activities of the body are designed to protect and care for the brain. Today, as I mentioned in our first lecture, brain life and death is the definition of death. We no longer go by whether the heart is beating or not, but by whether the brain is viable, alive, and able to function. There's great argument. The neurosurgeons and neuroanatomists like to make the rest of us feel bad because they tell us that the brain is there to be nurtured by the rest of the body, that everything else in that body is only there for the brain. The bones protect the brain. The bones in the rest of the body also make the body move to get things for the brain, to protect the brain. The heart pumps blood and oxygen and nutrients to the brain. The kidneys detoxify the blood for the brain. Any function you can talk about is there for the brain, but without the brain, there's nothing. I ran into an interesting discussion when I was giving a little lecture to my son's 4th grade class on the nervous system. And I put this question out to the children and said, what would happen if I took Cameron's brain and I transplanted it into Mary Beth? I can't do that right now, but maybe someday we can. Everything was Mary Beth except this piece of 3 pounds of tissue. Who would this new person be? Would it be Cameron or Mary Beth? And every child in that class said, that would be Cameron. I said, but it looks like Mary Beth. They said, but it would be Cameron. But it's got Mary Beth's physical abilities. Everything about this is Mary Beth. And they said, no, it would be Cameron. And they're quite right. When you get down to it, you are your brain. All your experience, your memory, everything is in that brain. And there's nothing else but the physical accoutrements that go along with the body for the care of that brain. So these little children were right. My college students gave me more of an argument. They didn't agree, but I think the 4th graders were right. There's a very interesting book which is now somewhat old, but still available that you might be interested in, by Roger Penrose, who is from England, and he wrote a book called The Emperor's New Brain. He's a neurophysiologist, and he discusses the brain versus the computer. Much too big a subject for us to get into here, but I think you will find that our brain is an extraordinary computer, and we're not anywhere near matching the capabilities with what we have today. The brain is protected by a bony, rigid case, the skull. And that skull is perfectly formed. And when you open up a human skull in the anatomy room and you look inside, every vessel and convolution in the brain is imprinted on that skull. So it's tailored to be a perfect fit. It has 2 layers of bone in it, in between which are air cells. And what this does, its round shape, first of all, gives it protection, so objects would be deflected off the brain. And the 2 layers with the air cells give it great strength and great lightness. If this bone were to be solid, the head would be very, very heavy and difficult to move around. And in enormous animals, like elephants, number one, they have very big neck muscles, but they also have a great many air cells to help keep this light. Now, the brain is covered, and now I don't even have to tell you what I'm going to compare it to, but the dura and the pleura. And I want to show you the coverings of the brain because they're vitally important in both structure and function and in the disease processes that take place. The coverings collectively are called the meninges. You've heard of meningitis, that's inflammation of the meninges. And if we look at the first slide, this is a section taken through the front of the head called a frontal section. Remember in the first lecture, I showed you how you could take a plane and cut straight through the body this way. That's a frontal section, and this is what's happened here. The patient is facing us. We've sliced through the patient and taken away the face and are looking at the front edge of the back of the brain, or in this case, the middle. That's different from sagittal sections going through you this way or horizontal sections. We'll get back to more of those. So this is a frontal section, and we see on top the scalp with the hair, some rather thick skin, and then a hard covering over the bone, a fibrous tissue called the gallia aponeurotica. That means a surface attaching to the muscles of your scalp. And here's the bone, the hard outer layer, the inner layer, and here's that bone which has all the air cells in it. Now, right directly under the bone, the first layer you come to looks like the looks like the parietal pericardium. It's kind of grayish brown, and depicted here in green, however, is the dura mater. The dura mater means hard mother, a hard protecting covering. That's what the mother refers to. And the dura covers the outer brain. And also, if you look in the middle, it comes together in the middle so that it actually also separates the two halves of the brain in a continuous layer. This is called the falx cerebri, and it just separates both cerebral lobes. The dura encases the entire brain. Beneath that is a very, very fine layer, and this is called the arachnoid. Arachnoid means spider-like. And if you look at these little fine projections going across between the arachnoid and the brain, that's where it gets its name. It looks like little spider webs. In the arachnoid are where many of the vessels are carried. Here's a vein and an artery shown schematically all throughout. Lots and lots of vascularity there. And the next layer is called the pia mater. The pia mater means the delicate mother, and it is the covering of the brain, and as I'm sure you all already know, it's analogous to the visceral pleura or the visceral pericardium. It's intimately attached to the brain. If you tear it off, you will tear the brain. The brain, by the way, feels like jello when you hold it. The brain, unfixed, is very loose and floppy and requires all this supporting structure to keep it in place. It's very, very jelly-like and dense because it's full of water. Now, these coverings have a lot of physiology as well as just physical protection. For example, there are these things called arachnoid villi, singular is arachnoid villus, that project up into cavities which are called sinuses. This is the superior, meaning on top, sagittal, meaning midline, remember that term from the sagittal plane, sinus. A sinus is a swelling or opening in the brain, in any structure. Here in the brain, it contains a fluid called cerebrospinal fluid, CSF. The CSF is a fluid that envelops the brain, and I'll talk to you more about it, but it is produced by the arachnoid villi. And these villi secrete the secretions, the fluids that bathe the brain in the cerebrospinal fluid. It's reabsorbed through dural sinuses, and then it has its own complete circulation, and there's actually a barrier. The function of the cerebrospinal fluid is to provide mechanical protection of the brain and the spinal cord. That's why cerebrospinal. It provides buoyancy. This floppy, jelly-like brain would get flattened against the bone, so it provides buoyancy for the brain. It suspends it, protects it, brings in nutrients to the brain, and enhances the escape of all the waste products, and makes the internal milieu, Claude Bernard's internal milieu, perfect for the brain, which happens to be different from the body. There are slight differences in the chemistry, the pH, the osmolarity, the concentration of solvents. that are in the brain that are different than the body. So there is this thing called the blood-brain barrier that separates them. And we have to be very cognizant of that when we talk about giving drugs, because certain drugs go in the bloodstream, get to all the tissues, but won't cross the blood-brain barrier, and others will. And when we design drugs for patients with brain problems, we want to design them so that they will cross that barrier and get into the brain and help the patient. Let me come back to the coverings and go clinical very fast here, because some of the most important and common things that happen to us happen to the brain coverings. If you get a fracture of the skull, a crack in the skull would be no worse than any other bone broken. In fact, they're rarely displaced. You don't have to set them. They have a good blood supply. They heal quite well. The problem is that if you look again, the dura is right against the bone. When the bone breaks, you almost certainly have a tear in the dura. This brings us to basically three kinds of problems you can have depending on where the injury takes place. Three clinical situations, and all with different complexes that we have to look at. When you get a tear in the dura, you will almost always tear some blood vessels too. And we get something called a subdural hematoma. You've probably heard about that term. It's very, very common. Subdural hematomas are collections of blood. Oma means something that has mass. Heme means blood. Hematoma is a mass of blood that's under the dura. You can also get an epidural hematoma. Now let me talk about that first. Epidural, we have dura here, we have a tear, and we have blood between the bone and the dura. Usually we've torn one of the connecting vessels. The epidural hematoma happens when you have a blunt trauma off into the skull, cracks the bone, tears the dura, and on top of the dura you get this collection of blood. The blood by itself would mean nothing because it could be absorbed by the body. However, it usually starts pressing. It expands and it starts pressing down. And where's it going to press on? It's going to press on the brain. It's the only place it can go because the bone is rigid. The epidural hematoma has a very classical syndrome associated with it. Patient is hit in the head, let's say with a club, or falls down, cracks their head. They are momentarily unconscious. We don't know why that happens exactly, but it seems to be the whole brain letting off an electrical depolarization and having to start again. But we're not actually clear about the physiology of unconsciousness. Anyway, the patient is knocked unconscious. We call that a concussion. If the patient is unconscious, comes back again, they have been concussed. And then they're clear for a while. They know who they are, where they are, and what happened. Some short period of time later, they start becoming unconscious again. What's happening is the hematoma is expanding. The first thing was the injury knocked them out. It's a KO. Then they wake up. Then the hematoma expands, pushes down on the brain, and starts causing brain damage and unconsciousness. The hallmark of the epidural hematoma is the lucid interval. The patient was awake and talking and then got bad again. And you can almost make that diagnosis like that without an x-ray. Of course, we wouldn't dare today. We get the x-ray, CAT scan, MRI, get the neurosurgeon in, and you go in and actually operate on that patient, remove the hematoma, sew up the dura, fix the fracture, and if you do it quickly enough, those patients do very, very well. The second one that you've probably more likely heard about is a subdural hematoma, and that's underneath here. This happens from trauma, but it happens particularly in the elderly. Because if you look at this section again, the brain in the elderly starts to shrink a little bit with advancing years, and it puts the vessels that are in here under tension. There's a bigger space between the dura and the arachnoid area and the brain. So when that patient gets an injury, blunt trauma to the head, often without a fracture, they get a tear in there from one of those vessels and they bleed under the dura. Now, often, initially, in fact, in most cases, those patients have no immediate symptoms. They are unconscious for a second and then they're fine, but they don't go back down again. Later on, weeks or even a month, it happened to my mother a month later, after a very minor injury, they start going into coma. They become disoriented, they start staggering, and eventually they're in coma very quickly. What's happened is their small hematoma has aged, and it has started to break down the products of blood, and it becomes a very hyperosmotic solution, a solution full of dissolved material. And because of the laws of fluid, water is sucked into that hematoma, and the hematoma rapidly expands and again pushes down on the brain. Now, the first surgical operation, or neurosurgical operation, probably that was ever done, was the release of a subdural hematoma. There have been primitive, prehistoric sites where brains have been found and skulls intact, where holes were made to relieve subdural hematomas. Drills were drilled through the brain and the skull, blood let out, and actually some of these patients survived, as evidenced by the fact that they had healed wounds on the findings of the archaeologists. I have in my possession some old instruments from over 100 years ago of hand drills that were used to drill through the brain, and today we do the same thing. We drill through the skull, put a catheter in, drain the hematoma, make sure the bleeding has stopped, and those patients usually do quite well. That's a subdural hematoma. The other one we have is subarachnoid, and what the subarachnoid hematoma is, it's usually from blunt trauma, and because the pia is torn, it's usually an intracranial bleed. It happens inside the brain. There's really nothing you can do about that. Just as if you had a stroke. Whatever damage is done. Doesn't usually do you any good to go after it because you'll just damage more brain tissue most of the time. There are exceptions. And so those are the three types of hematomas, very common, a big source of morbidity and mortality, and we have to treat them in their various complexities in different manners. Usually very good results if handled quickly and expeditiously. Let's go on to the cerebrospinal fluid and where it circulates. And we have taken you now from the blood-brain barrier, from the cerebrospinal fluid as a function of buoyancy, protection, and nutrition, and move on to the ventricles. There are in the brain, just as there are in the heart, ventricles, which means little belly. Only these ventricles are very, very different. Here are two views of the brain. This top one is taken from the side. It's as if we've removed the skull and all the coverings of the brain, and we're looking at the bare brain and the spinal cord. This bluish depiction of the ventricles is really an artist's sketch. It's as if we had X-ray vision and could look into the brain. Those ventricles are inside the brain. You cannot see them from the outside. And here they are viewed taking a look straight from the front to the back. They look like ram's horns, if you can see that. The ventricles have two components. There are the lateral ventricles. They're there and seen best here because they're lateral to the midline. They have a horn that comes down and goes into the temporal lobe, better seen here. And then they communicate with a midline structure called the third ventricle. They communicate with a fourth ventricle, and then a very small opening that goes all the way down the middle of the spinal cord between the cerebellum and the brainstem, and then down into the rest of the cord. This again is a cushioning effect. It also gives nutrients to the inside. It's a safety mechanism that gives the brain tremendous protection from trauma, which it needs. There are disease states in which circulation of the cerebrospinal fluid is impaired, usually in infants, and it can't get out. And the fluid is made in excess, and this leads to a disease called hydrocephalus, water in the brain. It is a purely mechanical problem. You've got to get the water out of the brain, and a hydraulic engineer named Holter had a daughter with this problem before doctors knew what to do, and he said, well, he looked at it as a simple physical problem of engineering. We need to find a way out, and he developed a valve called the Holter valve, which he designed, manufactured, and brought back to the neurosurgeons, and they made it surgically correct, and they put this one-way valve into the ventricles through some inert brain tissue and lead it out into the circulatory system, and this fluid can now drain out into the rest of the body and relieve the pressure on the brain. It has to be done early enough so that brain damage hasn't occurred. Not a very common disease, but one that's certainly distressing because it occurs in a newborn baby. The next thing we want to look at is the blood supply to the brain, as we will look at the blood supply to almost everything else we study. This is a picture of the basilar portion of the brain and shows the very, very rich blood supply. The brain gets its primary supply from two arteries, the right and left carotid arteries. This is the right because we're looking up at the brain from the bottom. The brain is lying on the table, top down. This is the front, this is the back, this is the right, and this is the left. So the two big carotid arteries actually come into the body from the arch of the aorta. The common carotid goes up into the neck. It branches into two branches. One goes to the face, the external carotid, and the internal carotid has no branches. It goes directly into the head. Now, when it reaches the head, here it shows the carotid arteries cut. It has a very interesting distribution. It has this huge artery called the middle cerebral artery, and then it gives branches forward, and it has a circle of anastomoses. It's a very unusual kind of anastomosis. Remember, we talked about vessels of nearly the same size joining each other. This is called the circle of Willis, named after Dr. Willis. It has this anterior, meaning the front, communicating artery, a posterior, cerebral and communicating arteries here. And then it communicates with this very big basal artery, which comes up from the vertebral column. The vertebral arteries, two of them one on each side, join in the middle, come up, and then make this circle. So this is very, very great redundancy to allow for the possibility of any of these arteries becoming plugged. For example, if this artery became plugged here, blood could actually come all the way around this side and get out into this portion of the brain. It's an elegant system of backup. There are patients, however, who have an incomplete circle of Willis. One of these communications is gone, and they're at much higher risk for strokes than otherwise the normal population would be. This is very important because unlike almost any other organ in the body, the brain doesn't have any room for metabolic reserves, like the storage of glucose in the liver. The brain has nothing but what's going on at that moment. Whatever the blood supply brings in the way of sugar, proteins, fats, and so on to the brain is there. There's no reserve. Its metabolic rate is so high that it needs a constant supply. So a loss of blood flow to the brain for any period of time, a matter of seconds, will result in unconsciousness, and in a matter of minutes will cause irreversible brain damage. We usually think of about 4 minutes as being the maximum that the brain can go without any nourishment before you get irreversible damage. Remember, we talked about fainting, how that might help restore the circulation to the brain by having the pressure gradient eased when you lie down. Drops in glucose can cause convulsions, seizures, can cause confusion, unconsciousness, and death. And so we need to assure, and evolution had to assure, that this would be protected at all times. There's an interesting reflex we have. I'm going to be telling you about the advanced brain, our modern brain, and our primitive brain. We have a brain that evolved from lower animals, so we have all the parts of the primitive brain still there. And part of our primitive brain, we've discovered only in the last few decades, has a reflex called the diving reflex. If you look at seabirds and animals like seals that dive in cold waters and go to great depths for long periods of time, the way they're able to do that is they have a reflex that once they hold their breath and go down in cold water, their brain shuts off the circulation to every part of the body that doesn't need it, and only allows circulation to the brain and minimally elsewhere. It turns out we have one that's similar, not as effective, but very similar. And it came from our primitive brain. We have found out quite by accident that patients, especially young children who have drowned in very cold water, have been resuscitated and without brain damage after a much longer period of time than we would have thought possible. So these young people were in the water maybe 20 or 30 minutes. I've heard of one as long documented as 30 minutes. Underwater, no respiration, have been brought back, the heart restarted, and the brain had no brain damage. Now, we would normally say 4 minutes is the outside, maybe 6. It happens in adults, not as much protective length of time in there, but still this diving reflex apparently is still present. And that's why doctors in an emergency situation will be more aggressive if the patient has drowned in very cold water than, let's say, in tropical waters where this reflex probably doesn't happen. And we have brain damage a lot earlier on. There's another part of the brain which is kind of interesting, and Carl Sagan wrote a lot about this, the astronomer and scientist who did a lot of deep thinking. He believed, and I think there's a lot of logic and other things to make us believe this too, that we maintained our primitive brain in the modern brain, that we still have fears that have nothing to do with reality and that have not been learned. If you put a baby that can just about crawl or sit up on a glass table, the baby has a startle reflex, will put out both hands and pull back as if he's trying to prevent a fall. Now that baby has no experience with falling. Yet in our primitive days, when we slept in trees, we evolved animals who slept in trees. That was one way animals could die easily, falling out of trees, falling off heights. And we believe that these reflexes, these startle and preventive reflexes, may be left over from that. We have an inborn fear of snakes and reptiles. You show a 1-year-old a snake, and you don't have to tell it that it's harmless or it's harmful. That child will be afraid of the snake. We have to consciously teach them that maybe there's nothing to be afraid of. But reptiles were the big animals, the big enemy when we were in the primitive stages of life. And Sagan believed that, again, that's a leftover instinct to avoid reptiles. He believes also, and I enjoy this one, that our dreams have nothing to do with what Freud and Carl Jung told us about. Maybe he's right, maybe he's wrong. He thought that we dream about every hour and a half during the night. And he believed that nighttime was the most dangerous time for primitive man. That was the time somebody was going to come along and eat you. And that we were awakened by our brain about every hour, hour and a half to make sure nobody was out there getting ready to kill us or eat us. And I have no reason to doubt him. I think Carl Sagan was a great mind, and I enjoy that explanation. So I'm going to move on now to something probably a little less exciting and talk about the anatomical divisions of the brain. There's a lot of new words in here, a lot of vocabulary. I don't want you to write them down or study them or memorize them. We're going to come back to them over and over again. Just kind of pay attention to the big picture, see where things are, let the words slip in and out, and the ones that we need to talk about again will come back. We are born with our brain very, very big. The brain is highly evolved even as we emerge from the mother at birth. That's why babies' heads look so big. That's why delivery is so difficult for the head, not the rest of the body. The baby's brain is disproportionately large when you consider the rest of the body's development. It is born with almost its complete complement of neurons. Those are the nerves that function in motor sensory integration. We've always thought we get no further neurons. It's now thought we get a few, but not many. And so what you're born with is what you get to keep, and we can't replace neurons right now until stem cell research allows that. Right now, you are stuck with what you've got. So the baby's brain is already very big. It has not finished its insulation, and that's why the babies have abnormal reflexes that would be pathologic in an adult. Because they don't have all their insulation wrapped up yet. That happens over a period of months and years, and that's part of the maturation of the baby's brain. Their eyes are also out of proportion and very big relative to the growth of the rest of the body. That's why they have such deliciously beautiful eyes. When you look at a baby, their eyes are very big. They actually are big compared to their face. So let's take a look at the brain now. And we're going to start with the primitive brain and work our way up. And it's interesting, the lower centers of the brain are the lower portions or more primitive, and we work up to the top of the brain where we have the most advanced parts of the brain. We're going to start now with this picture and give you just the positioning of the brain in the head. Here is the spinal cord. And this part is called the brain stem. It's as if you're looking at a flower. And here's the stem, and then here's the flower above. And then if we take a closer look, we can zoom in and see circulation of the fluid around the brain that encases all the different parts of the brain. You can see it's completely enveloped in cushioning fluid. Now, coming back and looking a little more closely, the first part of the brain and the stem that we're going to look at is called the diencephalon, and it begins at something called the medulla oblongata. Medulla means something central as opposed to the cortex, which is the outside, and the oblongata is because it's skinny and long. This is where the brain exits the skull. All the information is just below here is going to exit the skull and go to the rest of the body, with the exception of cranial nerves, nerves that go to the face and head primarily, and which we're going to spend a whole lecture on, so I won't go into them now. Every piece of information that goes to the body from the brain has to get out through this little hole. Even sensory feelings in the scalp don't come out of the top of the brain. They go out, down to the neck, and back up on top. So this little hole, actually it's called the big hole, the foramen magnum, is where everything comes out of the brain. In the medulla oblongata, by just way of review, we look at the exit of the last 5 cranial nerves 8, 9, 10, 11, and 12. Let those go in and out your ear. And also centers that regulate respiration, primitive functions, the respiratory center, sneezing, coughing, hiccuping, heart rate. We also have the crossing over of motor and sensory nerves in the medulla oblongata. There are certain nerves that switch sides, start at one side of the brain and cross over and work on the other side of the body. I have no idea why that happened in evolution, but it happened. And so when you get injury to the right side of your brain, you may have paralysis on the left side of your body. We talk about the ipsilateral side, meaning the same side, and the contralateral side, the opposite side. Moving up the brain, we go to something now called the pons. The pons is called the bridge. And this bridge controls more cranial nerves, some of the upper ones, nerves 5, 6, 7, and part of 8, which again we'll talk about later. And it's a place where motor and sensory tracts converge. The right and left sides of an organ called the cerebellum run through here. And the cerebellum is this thing that looks like a bun behind the brain. It's the center of intention and coordination. And this is the place where all the input from sensory areas of the body come to be coordinated and analyzed and then redistributed. What happens is that you are given a lot of information. I don't have to look down to know that I have one leg on the stool and one leg on the floor. I can feel it from sensation of pressure, and yet if my feet were in the air or if I were in a swimming pool floating, I'd know if my knee was bent or not, because I have organs of proprioception, spatial relation. In my joints, I have information telling me every second what every joint is doing. I know whether they're bent or straight. So I have touch, I have proprioception, I may have pain sensations, all coming in from every joint in the body all the time. While I'm awake, it's an unconscious coordination. I'm not aware of it unless I make myself aware of it. And this is all coordinated in the cerebellum. You can think of the cerebellum as moderating intent into activity. I want to do something, and I need to get all the information from my massive computer terminals, touch, feel, balance, everything, my eyesight. I can stand here and look at you and get cues as to my balance so that I can correct. But then if I close my eyes, I may have a harder time. I've lost some of the clues, but my inner ear will help me out. My feet on the floor will help me out. All this happens in the cerebellum. And lots of diseases that occur with balance and coordination happen because of the cerebellum. And we would never be able to do things like diving. An ice skater would never be able to jump, twirl in the air, land on one foot and skate off without a perfectly functioning and advanced cerebellum. We are learning now that the cerebellum actually may control much more than this. If you take, coming back to the picture, look at the cerebral hemispheres, very, very big. If you take this cerebellum and squash it out flat and line it up with the cerebral hemispheres, it's actually as big as one of them on the cortex, on its surface. So apparently there's much more room in there than we originally thought, and it has a lot to do with other things such as memory, things we never thought the cerebellum did. It also contains areas that have to do with more primitive reflexes, ones that we'll get into later. We move up then and look at the next level of the brain in the diencephalon, which is called the thalamus, and the hypothalamus, which means below the thalamus, and a gland called the pineal gland. The thalamus is the major center for receiving reflexes and relaying reflexes, for example, for knowledge, awareness, coordination again of thought, emotion, and memory. The thalamus, very, very important area that coordinates and integrates things that don't have specific locations in our brain. We can go ahead and do things to the brain, ablate areas of the brain. If we take out a place that controls function of your arm's not going to move. But we can't get rid of memory. Memory is holistic. Memory is like a hologram. I don't know if you know about holograms, but basically I took a picture of this room through a camera, and I cut the negative in half and made a print. Everybody on this side of the room that I cut out wouldn't be on that print. Only this side would be. But if I take a hologram and I cut my picture in half, I'm still going to have the whole picture. It's just going to be smaller. And if I cut it again, I'll have the whole picture. Every part of that picture is in every part of the hologram. And memory is like that. Memory is holistic. Memory carries the whole brain's, the whole brain carries the memory, and it's very hard for you to cut out a part of the brain and get rid of one kind of memory. Now, memory is very complex. Short-term and long-term memory, very, very different. And the studies on that are really almost in their investigative stages. We're learning more and more about memory all the time, and things that we thought were true at one time in our life are no longer considered true. So getting back to this part of the brain, this very complex mid-center of the brain, we have the pineal gland, which is down in this portion of the brain right there. Very tiny. It has to do with maintaining our body clock. It secretes melatonin, which has become a very popular drug lately, which is a sleep inducer. It sets the internal biological clock. We also have an area of the brain below the thalamus. The thalamus, as I said, gives you this coordination for acquisition of knowledge, of new knowledge, putting it into the files, awareness, knowing that you're conscious. emotion and memory. This is really hard stuff to quantitate. How we quantitate consciousness, for example. How do we know we are living? How do we know we are breathing? This is something that we don't know if other animals have, because we have no way to find it out from them, except by looking at their brains sometimes and seeing if they have similar structures. We know lots of the higher mammals have very similar brain structures, lower ones do not. Now, in the hypothalamus, we relay reflexes to areas related to smell. We're getting into very primitive stuff here again, too. Also, we get hormones that regulate the pituitary gland. I'm going to leave the pituitary gland for later when we talk about those hormones. The hypothalamus is a major regulator of homeostasis. It's going to correct, make changes in osmotic pressure. It's going to change hormone concentrations. It's going to take care of blood temperature. Remember how our body could open up the vessels on the skin, capillary beds, or constrict them depending on the situation. This is all coordinated in the hypothalamus. I want to talk for a minute now, too, about its functioning in the autonomic nervous system, because there's a whole part of your nervous system we'll get to in the next lecture that we don't control. Things like intestinal movement, the urinary bladder, the heart rate, and so forth. They're all controlled and coordinated through the hypothalamus. It also has some effect on emotion, aggression, pain, pleasure. It will regulate primitive responses such as thirst, hunger responses, and controls those things that are very, very basic that we never even think about. Our rhythms, our wakefulness, our sleeping are also controlled in the hypothalamus. So this very important area that we don't think much about, but certainly is on functioning and coordinating. And this computer that we call our brain is really has lots of information, a lot of it that we would think, if we had to think about it, our mind would be totally cluttered, unable to do anything else if we didn't have some primitive part, such as this area right here in the lower portions of the brain, to do everything for us and do this coordination. This brings us to the cerebrum, the cerebral cortex, which is the outer layer of the largest part of our brain. We have, in proportion to our body, the largest cerebrum of any animal on Earth. Elephant's brain is bigger and so on, but still, proportionally, we have the biggest and the most evolved one. We can do more with ours than any other animal that we know of. We have, as I've said, about 1 billion neurons, and the outer layer of this cortex is called the gray matter. The gray matter is because that's the coloration of the cells, and it's become an eponym for someone's thinking processes. We say he's got a lot of gray matter because that's where the highest functions of our brain are, the brain that we associate with intelligence. Below the gray matter is the white matter, and this is because of the shiny myelinated sheaths, which I'll talk to you about a little bit later, that insulate the brain, happen to hang out, so that everything that comes from the top of the brain and goes to the bottom goes through insulated wires, just like copper wires with duct tape around it or electrical tape. And so there's a different color. The brain is separated by that falx, which I told you about, the continuation of the door that goes down in the middle. So our brain has some separation in right and left. But there is something in the middle called the corpus callosum. And that's this body right here. It goes transversely across the brain, and it connects both sides. Callosum just means hard, corpus body, and when you feel it, it's quite hard. It would break like a carrot in your hand if you broke it. So the corpus callosum has transverse fibers that take information and tell the other side what the other brain is thinking. The brain is divided into hemispheres and lobes, and after we get into our right and left hemisphere, we have a frontal lobe here, and then we have a parietal lobe along the wall laterally. We have an occipital lobe, and we have one that goes down into our temples, which is best shown on this slide, and you can see the temporal lobe coming down here right behind our ear. And this is the basic structure of the brain. You can see it's very convoluted, has lots of gyrations and folds, again, giving it more space and surface area so we can cram more function into a smaller brain. We'll leave that here today and move on to more of the functioning of the brain in the next lecture.